Hello,
Hello.
how are you?
Pretty good.
Great.
Did I turn it back on? I did. Okay.
Haven't seen you in a couple weeks.
Yeah, I was on vacation.
Oh, okay. That's good.
Went to Barbados.
Oh, wow, good.
Got a sunburn and a bruised knee.
Oh, yeah. Yeah.
It breaks up the winter if you can go somewhere in the middle.
Oh, yeah, yeah.
Last year we went to the physics conference in March
Okay.
in
Yeah.
Las Vegas.
Oh, all right.
That helped. And this year I'm just going to attend it virtually because it's $170 Canadian.
Yeah.
So, like, what's that? US? Like, not very much.
It's good. Yeah.
Yeah, I missed a couple of things. Well, what happened?
Well, we had actually some good discussions last week on some papers on biophysics. There were some physical networks and other. Actually, we had a paper on that set of papers you talked about, trijunctions.
Oh, yeah.
Yeah, they had a paper where there were like multiple. I guess you could call them like quad junctions and pentajunctions and things like that.
Oh, could you send me that or?
Yeah, I will.
Yeah, I'm sure there's more than trijunctions because
Yeah.
things move around a lot.
Yeah, I just didn't know like if you had run across like how deep you were into that literature.
Not that much, but knowing that there are junctions there instead of the meeting of cell membranes, that adds to the morphology of the surface.
Yeah.
So when you're doing a graphic design of the surface, and looking at its physical characteristics, it changes it. I don't know by how much because they're very tiny. The junctions are tiny.
Yeah.
Bradley, have you heard from Vahid at all?
Not this week. No, I mean he was here last week at the meeting.
Oh, okay, because he hasn't been answering emails.
Oh, I don't know. Yeah.
Oh, okay.
Unusual for him.
Yeah.
Well, I just wondered what was up with him too, because I sent him a couple of papers about mitochondria. I'm interested in the high voltage between the membranes in mitochondria and anywhere else. Just wondering if you could if that would disrupt or distort infrared light. As in, could you detect whether a cell is alive using your optical coherence tomography? Like, I wonder if you could do that. Maybe it's just too fine a thing to even to worry about. But
That's Michael Levin.
Yeah.
The bioelectric stuff. Yeah.
But anyway, that's what I was after. Did he know anything about that? Like, if it is a fairly high electrical gradient. So, question is it detectable as a distortion of light?
Yeah.
Because it would be nice to know if your cell was alive, if you could kind of detect that with optical coherence tomography while you were working with the cell. So
I recall doing a time lapse one weekend where at the end of the time lapse, we actually slept in the lab. At the end of the time lapse, it was clear that the embryo was dead.
Yeah. Yes, it's that sort of thing. Yeah, if you could just kind of tell whether you thought it was alive, that would be helpful. Yeah.
Yeah. So
actually, you want to introduce yourself?
sorry, I didn't want to interrupt the conversation.
Okay.
I am Pakhi Banchialia. I'm from India. I am currently a third year undergraduate student.
Which department?
I'm in artificial intelligence.
Oh, okay.
I'm from Amrita Vishwavidyapeetham College in Kerala. And yeah, I would like to contribute to your open source projects. Like last year, I wanted to try, but then I thought I wasn't prepared enough. This time, I have prepared myself, and I think I will be able to make some meaningful contributions to your projects.
No, that's nice. Yeah, I haven't gotten into AI myself at all. Yeah, I was going to, and then I got hit with a bunch of other things. So.
Yeah. Well, welcome to the group. So we haven't announced the GSOC projects yet, the Google Summer of Code projects, but we'll be doing that soon. Usually, it's going to be very similar to last year's project, but I'm assuming about like DevoWorm and some of the things that we did last year. Yeah, and so we'll be talking about that more in coming weeks. So welcome to the group. And in the group we usually talk about a lot of things. We sometimes talk about more biological stuff, sometimes more AI stuff. So hopefully you'll find this useful.
I'm actually interested in merging like AI with medical sciences because I feel like it has a lot to help in the medical field. I am currently doing like a research project with one of my mentors. It's about segmenting the facial features to determine the level of paralysis a person might have. So that project is in process right now. So currently we are able to detect like the how much degree of paralysis the person has. There are more aspects to the project. Like we will try to make a system where we can monitor how well they are able to exercise to improve their for faster healing process. Yeah. So
that's good. All right. So let's see. Last week we did a number of biophysics papers. I think 4 in total. We went through a bunch of different topics. We did something on networks and I don't know. We talked about a paper previously where they had these rods in a packing and they were moving around, and they were moving around their radius of gyration and kind of moving together. And this one was about building networks like that, where you have a node and there's a radius around which the node travels, and then you have a bunch of rods that do that, and then there's some connectivity there. They called it spaghetti packings or something like that, which I thought that was interesting. We did another paper on cell sheets and looking at the different junctions that the cells make during morphogenesis. So Susan, at one time last year, I think late last year, was reading some papers on trijunctions, where the cells come together in this threefold junction. And in this paper, they talked about 4 junctions and 5 junctions, and higher order junctions where the cells are migrating within the sheet and they're forming these junctions. And it has a lot to say about differentiation and some of the things going on there. So those are some interesting papers. And I'll send Susan a copy of the one on the junctions later. So and then today I kind of wanted to talk a little bit about something Dick sent me an email about over the holiday, and I didn't get I did get a chance to look over things, but we didn't get a chance to do anything else with it. And let me share my screen. So this is about lineage-independent differentiation. So Dick sent me this. He was really excited about this literature on lineage-independent differentiation, and this is something that you find largely in plants. Although we'll see some of the papers that I have in this folder and what they have to say about it. So and this is just like a very quick overview of some things I found doing a literature search. So this is just kind of the starting of a topical deep dive into this. So if there is something that I missed, it's probably because I didn't really do a very good job of looking. But so this is actually in botany. So I'm not really that familiar with botany relative to animals. So, but this is the Journal of Experimental Botany. And this is paper Cell Identity Specification in Plants, Lessons from Flower Development. So this is, again, in plants and, the way that development proceeds there. So this kind of talks about this idea of lineage-independent differentiation. Multicellular organisms display a fascinating complexity of cellular identities and patterns of diversification. The concept of cell type aims to describe and categorize this complexity. In this review, we discuss the traditional concept of cell types and highlight the impact of single-cell technologies and spatialomics on the understanding of cellular differentiation in plants. So that's like a typical thing that we might do. We summarize and compare position-based and lineage-based mechanisms. So they differentiate between position-based and lineage-based mechanisms. So I take it position-based are things that are sort of have positional information or something like that. And then lineage-based is where they have this, like something like C. elegans, where if the cell divides, it's in this lineage that ends up restricted to a number of fates. Those are the two mechanisms of cell identity specification, and they use flower development as a model system. So, and more than understanding ontogenetic origins of differentiated cells, so they want to know like kind of the origins of different cells that become other cells. An important question in plant science is to understand their position and development stage specific heterogeneity. And so they get into this about the action of crosstalk and external and internal signals. This converges on transcription factors that orchestrate gene expression programs. So this kind of talks about, but this first part talks about how to define a cell type in plants. So defining cell types is sometimes a tricky business. I know that, some of there are groups that are always trying to define like cell types, and especially if you're doing like machine learning of cell types or machine learning of cells, you might want to know, like, have a methodology for defining cell types. So we've talked about this before, where you have cell types that can be defined by their markers, their phenotypic characteristics, their shape. Sometimes you have a marker that's expressed by a certain type of cell. But that doesn't mean that that cell is, that all those kind of cells are the same. There's a lot of heterogeneity there. All that means is it's expressing that specific marker. So you might go in and look at the gene expression profile, maybe like a sequencing technology, and find a lot of variation in that cell. You might find some cryptic version of a cell type, but you wouldn't know that from the marker, maybe not even from the morphology. So there are a lot of different ways you can define a cell. So a cell type is classically defined by phenotype and function. We've added molecular signatures to that. Less than 20 major cell types are classically assigned in vascular plants. So this is from 1989. So this means that there's a very small number of cell types in vascular plants. And I'm not familiar with plants, but I don't know how many functions there are in plants that are, as opposed to animals. That'd be kind of an interesting question to ask. But at least in this case, as of 1989, we had less than 20 major cell types. So this count was probably based on looking at the phenotype and function instead of more of the molecular signatures. Because
Bradley, for animals, I've seen figures from around 250 to 7,000.
Yeah. Well, they say major cell types too, so I'm not sure how that compares with, but still, it's probably
it's probably an order of magnitude or two less.
Yeah.
Okay.
Those classical cell type concepts aim to generalize cellular identity. It may not entirely cover the complexity and cellular diversity of cells. So this is what I was saying with, you might have cryptic cell types or transitional cell types, and they just have one of those markers meet one of the criterion, and so that's where you get a lot of variation in the number. Let's see if I can get down to something that's informative here. So in very practical terms, this creates challenges. This is talking about some of the aspects of like gene duplication followed by sub and neo-functionalization, co-option of genes, and gene regulatory modules. So these are all the things that you find in across species and plants and in animals for that matter. So those are things that can add to this diversification of cell types. In very practical terms, this creates challenges. For instance, since many developmental control genes act in more than one developmental process, that can be a problem. So many genes controlling vegetative leaf development are also expressed in floral organs. So you get these genes that are involved in vegetative leaf development, and they might be duplicated and then co-opted for another process. And then that's where you have, so those copies of those genes do something different. Their activities are modulated to give rise to specific structures of floral organs. This often makes it hard to identify marker genes that are very specifically expressed in only one cell type or different stages. So again, this is the idea of using a marker to say this is this marker positive these cells, and so you're going to have some variation there. One might compare the problem of defining a cell type with that of the species concept, and so it can be difficult to distinguish cell types from states, and cells with the same identity may appear very different from each other in terms of gene expression profiles. So there. Oh, go ahead. Yeah.
Do they discuss the syncytial plants?
I don't see anything here.
Because a syncytial alga can have different parts that look like leaves and look like stems, etc.
Yeah.
So that the differentiation is actually subcellular.
Right. I'll make a note. Yeah.
The spectacular form or the what do you call them? The kelps. You can have kelps that are cellular,
yeah,
and which can be 100 meters long, or you can have kelps that can be 100 meters long and one cell.
Yeah. You mean like just the cell gets bigger?
No.
Oh.
One cell.
One cell.
The whole plant is one cell.
Oh, yeah.
Okay.
Yeah.
And yet it has different parts.
Right. Okay,
now
one cell.
It has many nuclei.
Right.
Okay. But the nuclei seem to stream around, so I don't know what they're doing.
Yeah. Well, yeah, I don't know if they talk about syncytial cells or, that sort of thing in here, but so they give some examples from plants, sort of talking about how cell types that together constitute the epidermis tissue, and they talk about some of the aspects of these cells. And then together the example of the epidermis shows that the cell type composition of tissues can be variable across organs in a plant, and the individual cell types can have different phenotypes and function. And then, of course, they can also cell type frequencies within tissues can be plastic and affected by environmental factors as well. So this is, and then they talk about cellular differentiation being reversible in plants under specific conditions. This shows kind of the diversity of plant or cell types in plants. So you get different types of cells. And then this is sort of the I guess the gene expression network for trichrome and non-trichrome cells. So this is, let's see, this is B. B is movement of R3 MYB functions as a positional signal for trichrome patterning. So this R3 MYB plays a different role in this expression network than it is here. So yeah, this is kind of what they're getting at in this review. So this talks about cell lineage versus positional signal. So the fundamental concept of epigenetic landscape visualizes cell differentiation as a ball rolling down a valley in a landscape that is sculpted by regulatory genes and their combinatorial activities. And so in the animal field, reconstruction of cell lineage history is often used to understand cellular differentiation programs. So that's these reviews here. A classical example is for utilizing the lineage concept in plants is stomata development, which is initiated from a lineage-specific stem cell by a series of defined asymmetric cell divisions controlled by consecutively acting regulatory factors and enforced by cell-cell signaling. But many experiments have shown that the developmental fate of a plant cell does not depend strictly on its lineage, but on its exact position within the growing plant body. In early studies, plant scientists utilized mosaic or chimera experiments to trace the lineage of a cell type by labeling cell clones with genetically phenotypic traits such as ploidy level or albinism. So these are distinct things for a plant, and tracking the mitotic descendants of marked cells. And so these studies have shown that plant cells do not follow strict lineages and their fates are not predetermined, but rely on positional information or cell-cell interactions.
Yeah, that's true of animal cells too.
Yeah.
Except if you go back to Waddington, animal cells have what's called commitment.
Right.
But the commitment can depend on where they are.
Right.
So I don't think that's a good distinction.
Right. I was wondering about that because I'm like, well, I think that's also true of animals, but
the other thing, I don't know if they discussed it here, but the other thing that to keep in mind is that a plant like fucus has embryos.
Okay, yeah.
The embryos can develop, and so I don't know if there's any study of the development. Most people that I knew in fucus were just mostly interested in the egg itself.
Right.
So I don't know if any lineage work has been done on fucus.
Yeah. So this is kind of what they're getting, like, at least in this paper, this is what they're getting at for lineage independent development. So I mean, we find positional information dependence or whatever you want to call it in animal cells as well. And in fact, we know that in say like mammals, it's kind of hard to construct an exact lineage tree from developing cells in an embryo. Just that they're not always the same cell, that they kind of proliferate a cell type, and then those cells are get signals from their neighbors, and they kind of determine the cell type. So this is not something that's unique to plants, but of course, in fitting it into the epigenetic landscape, that's not necessarily here nor there. It's just kind of a different way of doing it. And so they talk about this sort of positional information or this perspective where plant cells may alter their identity when positional signals are changed. This is exemplified by observations on wound healing and in redifferentiation. So sometimes you'll get dedifferentiation in certain contexts where there's regeneration or wound healing. So cells of a certain type, like a muscle cell in animals, will dedifferentiate into like a stem cell state or a precursor state, and then redifferentiate as it divides a little bit and redifferentiate. So it replenishes that cell type in that region, and you can get that sort of process as well. Now, this kind of talks about this in terms of plants, though, and they kind of go through some of the aspects of positional dependence. So yeah, and then the rest of this, a lot of this paper is going to be molecular stuff that I'm not going to get into. So let's go to the other papers. This paper here is let's see. This paper here is a little bit. This is actually in animals. So this is in silico analysis to explore lineage-independent and dependent transcriptional programs associated with endothelial and neural differentiation of human-induced pluripotent stem cells. So again, we have this term lineage independence in animal cells. And so this is where they do some experiments here with actually with iPS cells. So they want to understand the endothelial cell type or the cell phenotype. They want to sort of get a better handle on the underlying molecular basis of this process. So in other work, they previously reported that the generation of iPS cells from human umbilical vein endothelial cells can be done. There's also the differentiation of resulting HIPS cells back to endothelial cells. So you can take a cell from human umbilical vein, they're endothelial cells. You can turn them into these stem cells that are induced, and then you can differentiate them back to that cell type. So, this is again where you give them, in cellular reprogramming, you'll often introduce, genes that are overexpressed, genes that will allow a cell to become a stem cell from a differentiated cell. And then when you redifferentiate the cells, you're guiding, it's a little bit less direct. You're guiding them down a certain pathway with maybe putting other cells of the same type in the culture or using a conditioned medium. So this is kind of how they approach this in animal studies. And so those cells then are lineage independent. But, maybe not, because they actually, the resulting stem cells are pluripotent. But it's, we haven't really looked at the sort of the range of cells that you can get. Usually in an experimental setting, you might take like a skin cell and differentiate it maybe into a muscle cell or back into a skin cell. We don't really know what the full range of fates are for that pluripotent cell. And so we know that pluripotent cells can differentiate back into a range of fates, but we don't know if given the source cell, what the full range of things are it can differentiate into. So this is, I guess semi-lineage, semi-independent maybe is a good way to put that. And so here we explored the same arrays to gain insight into the gene alteration process that accompanies the establishment of endothelial versus non-endothelial neural cell phenotypes. We compared the expression of genes that code for transcription factors and epigenetic regulators for this HIPS target phenotype. Our in silico analysis, which means they did some bioinformatics, have identified cohorts of genes that are similarly up or down regulated in both lineages, as well as those that exhibit lineage-specific alterations. Based on these results, we propose that genes that are similarly altered in both lineages participate in priming the stem cell for differentiation in a lineage-independent manner. So in this case, what they're looking at gene expression, and they're identifying different genes that are upregulated and downregulated. These exhibit lineage-specific alterations, and what they're just basically saying is that there's no lineage dependence that they can see in their study. Whereas those that are differentially altered in endothelial compared to neural cells participate in a lineage-specific differentiation process. So this is where you have cells that are differentially altered in endothelial cells. Those are different. Those participate in a lineage-specific differentiation process. So depending on the cell type or the original, or I guess it's some sort of, they're doing these alterations, and depending on what kind of alterations are made. Yeah, so they're actually they're doing this, working from this neural NN-diff cell lineage that contain both neurons and astrocytes. So the one that contained neurons and astrocytes actually had a different pattern than the ones that the source cells that just contained the HIPS cells, or the endothelial cells. So that's interesting that they're able to make that distinction between lineage dependence and independence. And, it's just maybe a matter of what it can differentiate into. So, maybe in neurons or in neural precursors, you need to maybe achieve a larger range of cell types. And in endothelial cells, maybe that's not so important. So I don't know why that would be the case. But in addition, we identified distinct cohorts of transcription factors and epigenetic regulators, whose alterations correlated specifically with the establishment of endothelial versus non-endothelial neural lineages. So that's in animals, and so that's the way they approach lineage independence there. This paper here is talking about stem cells. So this is division independence differentiation. So this is where we're independent of division, and we're talking about differentiation. Mandates proliferative competition among stem cells. So this is where you get different stem cells that proliferate at different rates. And so this is, I think, again, maybe in ants and Drosophila. So this is, again, in animals. So this talks about a little bit about these different cell types. Cancer-initiating gatekeeper mutations that arise in stem cells would be especially potent if they stabilize and expand an affected stem cell lineage. It is therefore important to understand how different stem cells organization strategies promote or prevent variant stem amplification or stem cell amplification in response to different types of mutation, including those that activate proliferation. So they're looking at are these gatekeeper mutations that do things. Sometimes they initiate cancer, sometimes they may initiate proliferation, which is a part of how cancer spreads, and so forth.
Bradley?
Yeah.
Stupid question. Plants are known to have tumors.
Yeah.
Do those tumors spread?
What is the tumor again? Or
do they spread like that?
Oh, I don't know.
I've never heard of metastasis in plants.
Oh, in plants. Yeah, I don't know. Yeah. I'm not sure.
I mean, if we get cancer, what kills you is usually it spreads throughout your body.
Right.
But I've never heard of that happening in plants.
Yeah, I don't. Yeah, I'm not familiar with that.
Okay, we'll throw it out as a thought.
Okay, so then stem cell numbers can be maintained constant while producing differentiated products through individual asymmetric division outcomes. So this is where we have, we're maintaining a constant number of stem cells. The differentiated products through individual asymmetric division outcomes are by population asymmetry strategies. Basically, if you kind of proliferate a certain stem cell sort of lineage, you could call it a lineage from like a single cell, and that cell's the mother cell when it can spread asymmetrically. So it's, this is where you might have differentiated products within the cell that can spread through like this differential reproduction. In which individual stem cell lineages necessarily compete for niche space. So, if you think about like a contained space, and you had a bunch of stem cells that started off as mother cells, and they started to build these lineages would proliferate across the closed space like a culture dish. Some lineages would outcompete others just because they're able to divide faster or do other things. And so that's what they're getting at here. We considered alternative mechanisms underlying population asymmetry and used quantitative modeling to predict starkly different consequences of altering proliferation rate. A variant, faster proliferating mutant stem cell, should compete better only when stem cell division and differentiation are independent processes. So this is where your processes, it doesn't follow that division implies differentiation. There's a different process there. So you might get fast division, and then you might get differentiation independent of that. But you don't have like division doesn't necessitate differentiation. For most types of stem cell, it has not been possible to ascertain experimentally whether division and differentiation are coupled. So this is where they can't know this for sure. However, Drosophila follicle stem cells, which are in Drosophila, it's a special type of stem cell population, provided a favorable system with which to investigate population asymmetry mechanisms and also for measuring the impact of altered proliferation on competition. So this is where, we want to see if we can find these asymmetry mechanisms and then measure the impact of altered proliferation. We found that from detailed cell lineage studies that division and differentiation of an individual follicle stem cell were not coupled, so that they are able in this model system to have a better handle on the difference between division and differentiation, and they found that those are not coupled in this system. We also found that FSC, or follicle stem cell representation, reflecting maintenance and amplification, was highly responsive to genetic changes that altered only the rate of follicle stem cell proliferation. This paradigm provides definitive experimental evidence for the general principle that relative proliferation rate will always be a major determinant of competition among stem cells, specifically when stem cell division and differentiation are independent. So this is basically showing that relative proliferation rate, or one lineage proliferating faster than or slower than the other, will determine the competition among stem cell lineages. And this basically says that there's this, it's division independent, that division and differentiation are independent. But it's not, I don't know if it's would be considered lineage independent. I guess it would be differentiation in this case might be lineage independent. But this is in a very specific model system. This is in Drosophila. And so, yeah, and I'm not going to get into the whole paper, but if you're interested, that would be something to look into. Yeah.
Does that paper discuss FOE? F-O-E.
F-O-E? Oh, you mean?
Yeah, that's her name.
Yeah, her name. No, Virginia Foe, right?
I don't recall the first name.
Okay. Let's see. I'll go down to the references.
She found that at certain stages of the Drosophila, the differentiation seems to occur in waves, which which may correspond to differentiation waves.
Yeah. Yeah, I don't see that. I don't see that citation
They
here.
didn't do it alphabetical.
Yeah.
Search.
Okay. Let's see. I don't know if I can search the way I have it here, but oh, I guess I can. Nope, I don't see it. No. But yeah, that's. I'll just make another note.
Okay.
So who was that? FOE?
FOE. Yeah, Virginia Folk.
Oh.
Her work was around 1993.
Yeah. We talked about her before in the group.
Yeah.
So. Okay, so this, all right, that's that paper. This one. Okay. So yeah, this is one that Dick took some notes on here. This is going back to plants. So we had two examples from animals. One, the original one was from plants. This one is going back to plants. So these are some notes here in highlighted text here, as you can see. The title was Directed Cell-to-Cell Movement of Functional Proteins. And then the question is, do transcriptional factors double as signal molecules in plants? So this is where they're asking about cell-cell communication and some of these signals and what they look like. So a couple of highlighted pieces here. In plants, cell fate is generally determined by position, not cell lineage. So this is, again, we talked about this, that this is also true in animals, but that's kind of maybe what they mean is it's predominantly determined by position, as opposed to in animals where you have cell lineages. But again, it depends on the organism you're talking about. In both cellular differentiation and organ development depend on intracellular signaling. So this is what we're talking about with signaling molecules and cell-cell communication. And so, but then, what is, the question is, what does that look like? What are these signals? A widely accepted paradigm from the control of gene expression during development by intracellular signaling specifies the release of small signal molecules by the signaling cell, perception by the target cell, transduction of the signal to the nucleus, followed by modulation of transcriptional control. So you have a cell that's sending a signal, and they're using small molecules. And then the target cell has to perceive that and recognize that signal. And then that signal has to be transduced to the nucleus from maybe like a surface receptor. And then there has to be some action, some modulation of transcriptional control. So that's what the model is. And of course, it doesn't have to follow that model exactly. We know also from synaptic communication in neurons that you can also have processes that go out, and you can have, in neurons, you have formal synapses where chemical signals are exchanged, and those can be quite complex, the number of receptors and the number of things released. And those can also, those don't necessarily affect the nucleus directly, but a lot of times they do. But so there's a lot of complexity in this communication channel. So that's what they're getting at with cell-cell communication. And so there's this directed movement of functional proteins. And so now, however, a new paradigm gaining attention proposes that transcription factors themselves can act as signaling molecules in higher plants, moving between cells to activate gene expression at a distance from the site where they were originally produced. So that means you can have a transcription factor that's expressed in the nucleus of one cell. It can be transported out into the cytoplasm. It can go to the membrane. It can then be released and dock with a neighboring cell, go back into that cell's cytoplasm, end up in the nucleus there, and initiate transcription in the same way it did in the first cell. So that's what they're saying here. This is a radical hypothesis, and they looked at plant viruses mediating intercellular movement of viral proteins and nucleic acids by physical dilation of intracellular plant organelles known as plasmodesmata. So this is something in plants that they've observed, but then generalizing it is a little bit radical of a hypothesis. So although Lucas's hypothesis for this type of trafficking of endogenous plant proteins was initially met with some skepticism, a major step towards its validation has now been taken by Sessions. He used genetic chimeras to demonstrate that cell-to-cell movement of a plant transcription factor results in the direct activation of target gene expression in cells adjacent to those in which the transcription factor was originally produced. So this, again, you might have two cells that are sitting next to one another from different cell lineages, but if they can make these exchanges, they can be regulated in the way that, maybe neighboring cells should be regulated or something like that. So it doesn't necessarily matter what their origins in a cell lineage were. It just matters what's being communicated between the cells. And so, you can see this in cells where there are a lot of examples of cell-cell communication. A particularly interesting example is the knotted1 mutant in maize. Knotted1, or KN1, is a homeobot containing transcription factor responsible for the maintenance of meristem cells in a multipotent state. So sometimes you get breakage and occasional loss of the dominant knotted1 allele. This shows that the KN1 protein expresses ectopically in the veins of a developing maize leaf and acts in a non-cell autonomous way to control cell division in the surrounding mesophyll and epidermal layers. So this is, again, an example of a gene where you get this kind of effect that's position dependent or maybe tissue dependent instead of cell lineage dependent. So there was a message in this chat here. These are some citations here. Virginia Foe. I think there are 5 here. So these are, again, kind of talking about some of these works here. Okay, so yeah, this example is from, they use a number of examples from plants. This is an example here, an image that shows the movement of a transcription factor in the meristem through the plasmodesmata. So this is LFY, the LFY transcription factor, which is in yellow up here. Let me see. It produced by the cells on the L1 layer can travel through the plasmodesmata to influence gene expression in the underlying layers, L2 and L3 of the meristem. So you can see this is the L1 layer at the top here. This is the meristem here. This is the LFY expression in these cells. And you see these like these kind of lines between the cells in the upper layer. This means that they're able to exchange LFY between those cells. But for now, the lower cells, L2 and L3, are blocked. You can't have exchange between cells of different layers. So the LFY transcription factor is exchanged across this upper layer. And then, so yeah, it says but it has to travel through this plasmodesmata to influence gene expression in the underlying layers, L2 and L3. So you see that L2 and L3 then can be influenced, but only through a different mechanism. And so this is kind of what they're talking about. So there's this, this sort of positional aspect to this. Yeah, that's that paper. Actually, I wonder if at the end of the paper they talk a little bit about some of these questions, open questions. Yeah, so questions as answers will be essential to our understanding include A, over what distances and at what rates do functional proteins traffic in developing plants? So these functional proteins, like say LFY, they traffic in a certain pattern. What is the sort of the range, the distance, the length scale under which they travel, and what rate do they travel at?
Now, Bradley, there's an interesting aspect to this, and that is that the cell wall at the middle of the meristem is very soft.
Yeah.
And it gradually gets harder. So not only do you get this transcription factor, but you're also getting propagation of stiffness.
Yeah, that's true. Yeah. And then, so B, which directions of movement are permissible for each trafficking factor? So we saw that LFY can be transmitted across L1, and then it could be transmitted across L2 and L3 in a semi-permeable manner. So, we don't know exactly what directions of movement are permissible. It might be that it's only permissible in one direction. It might be like a unidirectional thing where you have a concentric circle around the cell, and it just broadcasts out like that. That might be an interesting thing. And in fact, more generally, people have studied sort of cell-cell communication, and we kind of assume that things kind of get broadcast out from a cell, so everything around the cell gets the signal at a certain distance. So it might be like, at maybe 10 to 20 microns, you might get the signal, or maybe 5 microns, but it might be differential in terms of direction. So you might get a signal that only goes to the left over the right, and there might be different length scales within a single broadcast of the signal. So that's kind of interesting. That's an interesting question as well. C is, what are the factors that limit or promote the movement of specific proteins? So these are, again, it could be stiffness. It could be other factors that play a role. It could be other transcription factors, or it could be like migration even can be taken into account. So if we were thinking of building a computational model of lineage-independent differentiation, we might have to take, of course, the model of how signals are propagated into account, some of the structure of the tissue into account, and then maybe the physics of the tissue into account. And then last but not least, what are the precise developmental consequences of altering traffic patterns of transcription factors in meristems and developing organs? So what are the precise developmental consequences? Meaning, what does this actually do in the tissue? And if we knock that out, what is the consequence of knocking that mechanism out? And so that's something that they don't really cover here. They just kind of demonstrate it. So if we were to have to rely on cell-dependent differentiation, what would that look like? The final paper here is this developmental cell paper, single cell resolution of lineage trajectories, and the Arabidopsis stomatal lineage in developing leaf. So I'll just go through the graphical abstract here and kind of talk a little bit about this, what they're getting at in this study. So they ask a question: how do you build a leaf? And so this is a cartoon of a leaf, and this little black circle is this cross section here, where you get these different cells organized in these layers. This is the kind of where we have these different cell types. So this is a scRNA-seq model of the developing leaf. So this is the sequencing study where they do this this multi-dimensional projection, this multi-dimensional reduction of gene expression, and you get these clusters of different cell types by their gene expression profile. So you get these are mesophyll cells in this layer, and they correspond to a certain gene expression profile. You get vasculature, which is in this layer, and this is gene expression profile over here. So there's some overlap, but they kind of correspond to these different layers. you get the epidermis or the epidermal cells at the bottom and at the top as well. And you get these different patterns of gene expression. So they're actually separable. Some epidermal cells express genes here and some express genes here. And there's some overlap in expression. But the point is that these different layers express different have gene expression profiles that are different. And, how do you get to that point? So how do you build a leaf in these different layers, these distinct layers, and then get the cells to be coordinated in terms of their gene expression? So this gives us a revised model of epidermal stromatal lineage, where you get these differentiated cell types. You get a pavement cell. You get the stomata here, which is part of the cell. There's this process by which it kind of goes through. I guess these are two different pathways from involving these different genes, and they result in different phenotypes. And then this is a functional analysis of extended expression. So I guess this overlapping area here, where you get these this sort of overlapping gene expression, you're getting this wild type epidermis versus late stage loss of SPCH. So it affects the stomata, and this is so they're kind of going through. They're looking at the leaf cross section. They're looking at these different layers. They're looking at the gene expression profiles. Then they're looking at the pathways and how they produce developmental outcomes. And then they're looking at mutant phenotypes, and they're looking at when you have stomata versus no stomata. So this is actually pointing to a different paper. This is kind of one of these resource papers, they call them. And this cites a paper, Lopez-Anito et al. And so that's the actual study. This is kind of the in brief. So the question is, how do cells build developmentally flexible organs? And in this case, we're interested in the stomata and how those can be adapted flexibly. And so Lopez-Anito et al. Employ a single cell transcriptomics approach to uncover distinct models of cell differentiation within Arabidopsis leaf tissue. So these two pathways are these two modes of cell differentiation, along with revealing dynamics of cellular programs, the identified underlying cell heterogeneity within the epidermal lineage, and interrogated new roles for a core transcriptional regulator.
So, how does a plant get along without its stomata?
I'm not sure. But that's what this is a mutant phenotype, so it's probably not viable necessarily. But,
yeah.
So the highlights from this are that distinct models of cell differentiation and lineage trajectories define leaf tissue. So we get these lineage trajectories, which are important. Cell cycle regulators exhibit distinct yet overlapping expression profiles. So that's B. And then, so you have these regulators that are involved in, say, forming the stomata. They have this overlapping gene expression profile, and this can result in this mutant phenotype. C, flexible stromatal lineages exhibit cell states and exist along a continuum. So these stromatal lineage cell states can be, different in nature. They can take different pathways. They can result in different outcomes. And D, single cell resolution refine cell fate commitment decisions that yield stomata. So these are, we can look at single cells and their gene expression profile, and we can actually get into what these fate commitment decisions are. So, it's not just a matter of blindly following a lineage and having this deterministic expression, this phenotype expressed deterministically. There are these different pathways that get taken and decisions that are made by the cell. So this is, kind of argues that there's maybe the cell lineage independence is a little bit more nuanced than we thought just from the name. And so, this is, again, in plants. So we had some examples, 3 examples from plants, 2 examples from animals. Thank you, Pakhi, for joining us. Hope you enjoyed the meeting. All right, so do we have any questions or comments?
I'm not up on plants that much.
Yeah, the caveat here is I don't really know that much about plants, so.
Yeah. Bradley, did you run into anything? I'm trying to recall the guy's name. The guy who worked on the development of sunflower patterns. You know, the seeds are in spirals and they're
right.
Fibonacci sequence and whatnot.
Yeah.
He died young. I'm trying to remember his name.
I don't know. I didn't run across anything on that.
At any rate, I mentioned it in my book, Hierarchical Genome, and it appeared to me to be an example of differentiation waves.
Okay. Yeah, I have to look and see if I can find something specifically on that.
Yeah, I'd have to dig the book out and then get searching. Okay. Anyway, actually, I might be able to do that. But give me a second here. Let's see. Okay, now. Oh, Paul Green.
Paul Green, okay.
That's his name.
All right. I'm searching through the book, but it's very slow. Yeah.
I hit his name. I'm trying to see if I can get a reference.
Yeah.
Green. I've got a paper nearby. Then Green. In 1989. I don't have a reference yet.
Okay.
No point in waiting. This could take too long.
Okay. Well, I'll see if I can find something on that and report back.
Yeah.
Okay. So I'll look into that, see if I can find it, and report back. Yeah. So yeah, it was good. So thanks for attending, and talk to you next week.
Okay.
All right.
Take care.
Bye. Thanks.
Bye.
Bye. Okay, now I'd like to go over a paper on fluid flows in biology, which is a nice new paper that came out. Okay, so this paper is called Three-Dimensional Soft Streaming. This paper is on something called viscous streaming, especially with respect to the soft bodies of biological organisms. So we'll go through, this is a very mathematical paper. This is from the Journal of Fluid Mechanics. And so we'll just kind of go through some of the conceptual aspects of this. One of the last author on this paper is Mattia Gazzola. And this is the website for the Gazzola Lab. So this is They're based at UIUC. I think they're in the genomic biology building, and they're also in mechanical engineering. So this is soft creatures. This is an example of some of the things that they've worked on: soft creatures, cyborgs, biolocomotion in fluids, the mind in vitro, which I guess is like some of these organoid on a chip things, viscous streaming, which is what we're going to talk about, where they have this donut in a flow field, and they're going to talk about that. We'll talk a little bit about viscous streaming and some of the uses of it. So viscous streaming is an efficient rectification mechanism to exploit flow inertia at small scales for fluid and particle manipulation. So this is, again, rectification we'll talk about what that is. It typically entails a fluid vibrating around an immersed solid feature that, by concentrating stresses, modulates the emergence of steady flows of useful topology. So this is where a fluid moves around like some solid feature. It could be like a soft body, which would be an organism, like, say, like, for example, an organism moving through a water column. The fluid is vibrating around it, and there's a concentration of stresses during that interaction, and it modulates the emergence of steady flows and things that maybe propel the organism or buffer the organism during movement or something like that. Motivated by its relevance in biological and artificial settings, characterized by soft materials, recent studies have theoretically elucidated in 2 dimensions the impact of body elasticity on streaming flows. So we know that, like, if we have like a body of a fish going through water, it can have an impact on the streaming flow. If you have water streaming in one direction and the fish swimming upstream, there can be interactions between the body itself and the water flow. So this is, we know that the relative elasticity of the body, whether it's very soft or very rigid, can be impacted by these streaming flows. Here we generalize these findings to 3 dimensions by the minimal case of an immersed soft sphere. We first improved the existing solutions for the rigid sphere limit by considering previously unaccounted terms. We then enable body compliance, which means sort of the softness of the body, exposing a 3-dimensional elastic streaming process available even in Stokes flows. And this is a type of turbulent flow here. Such effect, consistent with 2-dimensional analysis, but analytically distinct, is validated against direct numerical simulations and shown to translate the bodies of complex geometry and topology, paving the way for advanced forms of flow control. So one of the reasons you might want to use something like this is for maybe like a biobot or a soft robot or something like that. So this study investigates the effects of body elasticity on 3-dimensional viscous streaming. Viscous streaming, which is the sort of the topic of what the hydrodynamic aspect of this is. Viscous streaming, an inertial phenomenon, refers to the steady rectified flows that emerge when a fluid oscillates around a localized microfeature. So you have an interaction between the flow and something in the flow, and there's this fluid oscillation right at that where it hits the feature, and there you go. All right, so viscous streaming, an inertial phenomenon, refers to the steady rectified flows that emerge when a fluid oscillates around a localized microfeature. So if you have a pipe and you have a flow going through, you have local microfeatures where there's an oscillation of the fluid when it comes into contact with that feature. Given its ability to remodel surrounding flows over a short time and length scale, streaming has found application in multiple aspects of microfluidics, from particle manipulation, and there are a bunch of citations here, and chemical mixing, citations here, to vesicle transport and lysis. There are citations. There's one citation, or two citations here. So vesicle transport, of course, is important in cells. They transport through the cytoplasm, or you get lysis, where you get the cell breaking apart. And so these are all important places where this is found. Recently, the use of multi-curvature streaming bodies has expanded the ability to manipulate flows, leading to compact, robust, and tunable devices for filtering and separating both synthetic and biological particles. And so this is in a microfluidic chamber where you might sort particles, where you might sort things, chemical compounds or cells. And so you can use these kind of, this sort of streaming, viscous streaming to do this. So more recently yet, motivated by medical and biological applications and building upon past theoretical studies, in the citations here, the effect of body compliance has been considered. And so this is where the object has some sort of compliance or softness or ability to sort of absorb a lot of these variations in the flow. With one of the studies yielding the first two-dimensional streaming theory for soft cylinders. So this citation talks about streaming theory for soft cylinders. Its major outcome is encapsulated in this equation here, where this term is the time average Stokes stream function, and R, for these two variables, are the radial and polar coordinates in the cylindrical system. So you have this characterization of the cylindrical system, and you have this time average Stokes stream function. This relation reveals an additional streaming process purely induced by body elasticity that is available even in Stokes flows where rigid body streaming cannot exist. So rigid body streaming is this term here, which is this term here. So we have this additional streaming processes here. It's additive to this rigid body streaming here. So you see we're assembling this streaming process, and we have the coordinate system here, and then this is our Stokes stream function. In this work, we extend this understanding to 3 dimensions by examining the minimal case of an oscillating soft sphere. We first present an improved theoretical solution for the rigid sphere case by augmenting the derivation of Lane with a previously unaccounted term related to vortex stretching. So they're characterizing our mathematical model, they're setting up the equations. So there are a lot of equations in this paper, but basically this is figure 1, this is the problem setup. You have this 3-dimensional viscoelastic solid sphere, and this has a radius of A with a rigid inclusion immersed in a viscous fluid. So this viscous fluid here, it's immersed inside this. This is the radius, and this is the elastic solid on the outside. And so this is, again, in 3 dimensions. In this study, we deploy a spherical coordinate system where the radial, polar, and azimuthal coordinates are represented. The sphere is exposed to an oscillatory flow with the far field velocity in the x direction along the axis of symmetry. And then 2-dimensional axisymmetric cross-section of the elastic sphere is shown here, where you can see some of these aspects of it. So this is the axis of symmetry along the x-axis, and there you go. So this is figure 2, and this kind of shows a little bit of the elastic sphere and streaming flow response. So we have our sphere here, and we have our flows. And so A through C, which are these images up at the top, these are the 3-dimensional time averaged Lagrangians for this model. This is the Stokes stream function depicting the streaming response at m equals 6 with increasing softness. So this is where we start off with an initial condition, and you have increasing softness as you go from A to C. This is the softer, so the parameters change as the material gets softer, and you can see that this flow changes as well. Blue-orange represent clockwise, counterclockwise rotating regions. So you can see that the blue and orange represent these different regions of rotation. And so then if we go to this figure D here, this is a description here of normalized DC layer thickness, which is the y-axis, versus the inverse Walmsley number. And this is, of course, a theoretical prediction, so we have a number of different conditions. This is the Lane prediction of Lane 1955, so this was the original model that we talked about earlier. This is the Riley model in 1966, so this is rigid body theory from Lane, this is rigid body theory from Riley. And so this is what theory predicts, and then the empirical observations are below here. They're actually getting a little bit different result from the theory. This is rigid body where cow is 0. This is elastic body where cow is 0.0125. And as cow increases, this function goes lower in an elastic body. So this is the normalized DC layer thickness. It decreases. You get this function that deviates from the original theory. And then finally in figure 3, we see this extension to complex bodies. So this is where we get our torus, that we have this ring that we can see it's interacting with the flow. So you get these vortices that occur along the boundary of this torus, and then you get these flow fields out here that are colored. So this is an extension to a complex body. Instead of just the sphere, now we have a torus that's interacting with the flow. Here we consider compliance-induced streaming in a soft torus, a complex shape entailing multiple curvatures and distinct topology relative to the sphere. Numerically simulated time-average Eulerian flow topologies for a torus of core radius r and cross-sectional radius a, with varying body elasticity cow. So cow is the varying body elasticity. And so we can see that the torus is pinned at the center of its circular cross-section by a rigid toroidal inclusion of radius 0.4a. All other physical and simulation parameters are consistent with sphere streaming. So you can see that there's a difference here in the way this torus behaves versus the sphere. So the conclusion, in summary, the study improves existing three-dimensional rigid sphere streaming theory. So this is an improvement upon the theory as we saw in figure 2. It expands it to the case of elastic materials and further corroborates it by means of direct numerical simulations. Our work reveals, in keeping with our previous work on two-dimensional soft cylinders, an additional streaming mode, accessible through material compliance and available even in Stokes flows. It further demonstrates that body elasticity strengthens streaming, or enables it at significantly lower frequencies relative to rigid bodies. So this is interesting because we can take, and this is largely thinking in terms of applications. You can design things with a very soft body. It has high elasticity, and it can interact with a flow in different ways than a rigid object would. And so you can exploit the design feature, say for something in a microfluidics context, where you want to optimize that process, or in some other context. And this is not pure biology or developmental biology like we talk about in this group, but this is kind of instructive as to like how some of these geometries and how some of these body physics might evolve or might benefit from certain types of differentiation. So this is an interesting question. I don't really have a good application domain for it in, say, developmental biology right now, but it is interesting to think about, especially considering that we talked about lineage-independent differentiation, and we talked about some of the aspects of physics of cell sheets and physics of different tissues on differentiation and maybe guiding differentiation. So it's a very interesting question. So they kind of talk a little bit about extending these theoretical insights maybe to geometries other than the sphere. And so these findings advance our fundamental understanding of streaming flows with potential implications in both biological and engineering domains. And so again, these streaming flows are very important structures in the environment, and they can have an effect on biological systems, very soft-bodied systems, and otherwise.